The nervous system. With just over 100 billion neurons, nerve cells, around 1 billion synapses, the connections between neurons, and hundreds of chemical substances that modulate the activity of this vast network, the human brain appears to be a system of unrivaled complexity within the living world. Thus, functions as complex as memory, consciousness, and language arise from the physicochemical properties of neurons, the circuits they form with one another, and the information they convey in the form of electrical signals. Functions of the nervous system. The nervous system has three main functions: sensory, integrative, and motor. One sensory function. It detects certain changes, stimuli, both inside the body, internal environment, and outside the body, external environment. Two integrating function. Enables us to analyze sensory information and make decisions about appropriate behavior. Three motor function. It can respond to stimuli by means of muscle contractions or glandular secretions. Divisions of the nervous system. The nervous system comprises two main divisions. One is the central nervous system, nerve axis. It comprises the brain and the spinal cord. The encephalon comprises the three organs located within the cranial cavity: the brain, the cerebellum, and the brainstem. The spinal cord is located within the spinal canal, which is formed by the vertebrae of the spine stacked one on top of the other. These parts of the central nervous system act as integration centers that analyze and interpret sensory information in order to generate motor commands based on the individual's experience, reflexes, and the conditions prevailing in the external environment. 2. The peripheral nervous system. It comprises the parts of the nervous system located outside the cranial cavity and the spinal canal, and therefore outside the central nervous system. These organs correspond to the various nerves connected to the brain or the spinal cord. The nerves that attach to the brainstem are called cranial nerves, whilst those that attach to the spinal cord are spinal nerves car ils emergent du canal rachidien. This communication network connects the organs and peripheral viscera to the central nervous system. Diagram summarizing the entire nervous system. Nervous tissue. The nervous system is made up of two main types of cell. Glial cells, which are collectively known as neuroglia. It accounts for 90% of all cells in the nervous system, and its role is to support and protect neurons. Neurons account for 10% of all cells in the nervous system. They perform the specific functions associated with the nervous system. Neurology. Nine times as numerous as neurons, glial cells make up the neuroglia. They comprise several cell types, with a wide range of functions, some of which are only just beginning to be understood. These are cells that support the nervous system. They are much smaller and more numerous than neurons. They are capable of replicating by mitosis. Their functions include: they play a role in nourishing neurons and removing waste products. They guide the migration of neurons, provide immune protection, insulate the axons of neurons. In the central nervous system, there are four main types. 1. Oligodendrocytes. These are the most common cells in the CNS. They are smaller than astrocytes and have fewer extensions. Their cell bodies are round or oval. Function: they ensure the myelination of nerve fibers, thereby enabling them to increase the speed at which the electrical signals they carry are conducted. 2. Astrocytes. these are star-shaped cells with numerous extensions. They are found in the gray and white matter of the CNS. Function: regulate the composition of the extracellular fluid in which neurons are immersed and also play a supportive role by contributing to their migration and growth during development. Very recently, it has been discovered that they are also involved in immune mechanisms. 3 are microglia. These are small phagocytic cells, macrophages, with few extensions. Function: they are responsible for cleansing the nervous tissue and ridding it of any intruders. 4 ependymocytes. These are epithelial cells arranged in a single layer, ranging in shape from squamous to cylindrical and ciliate. Function: they form the continuous epithelial lining of the cerebral ventricles and the ependymal canal. At the device level, we know of only one type represented by Schwann cells, named after the German physiologist Theodor Schwann, who observed them in the late 19th century. These are flattened cells arranged around axons in the SNP. Function: they ensure myelination within the nerves and provide support for unmyelinated fibers. Finally, it has recently been discovered that glial cells play a role in the formation of synapses and are capable of strengthening or weakening the mechanisms of synaptic transmission over time. The cells of the Neuroglia. Le neuron. As the basic unit of the nervous system, the neuron is the cell responsible for the generation, creation, processing and transmission of information. A neuron is a communication cell. Each neuron receives information from 100,000 neurons and sends information to 100,000 neurons. Neurons come in various shapes and are composed of the following parts: the cell body or perikaryon, the cytoplasmic extensions represented by dendrites, the axon, the cell body, perikaryon or soma. It has no definite shape. Its diameter ranges from 5 to 135 m, surrounded by a membrane called neurolemma. Contains a nucleus surrounded by cytoplasm known as neuroplasm, which comprises organelles such as lysosomes, mitochondria, and the Golgi apparatus. The cytoplasm contains characteristic structures found in neurons known as Nissl bodies. Cytoplasmic extensions. Dendrites and the axon are extensions of the cytoplasm that enable the neuron to interact with other neurons, interneuronal synapses. Sensory receptors, neurosensory synapses. Muscle or glandular cells, neuroeffector synapses. Dendrites. Dendrites are generally short, numerous and highly branched. Dendrites are not myelinated. They contain mitochondria, smooth endoplasmic reticulum, numerous microtubules and are rich in ribosomes, which enables them to synthesize some of their own proteins. Some of them sometimes have thousands of tiny cytoplasmic protrusions on their surface, no larger than 2 m, which are known as dendritic spines, and which are generally the site of synaptic contact. The axon. It is a long, slender, cylindrical extension that may be myelinated. It joins the cell body at a conical elevation known as the axon implantation cone. An axon is always a single structure, but it can branch out into collateral branches that enable it to transmit its messages in different directions. The axon and its extensions end by branching into numerous fine extensions known as axon terminals. The ends of the axonal terminals swell and become bulbous structures known as synaptic terminal buttons, which contain synaptic vesicles that store neurotransmitters. Its cytoplasm, sometimes referred to as axoplasm, contains no ribosomes. It contains some smooth endoplasmic reticulum, mitochondria, and microtubules. Classification of neurons. The various neurons in the nervous system are classified according to their morphological structures and their functions. Structural classification. This allows us to identify three types of neurons: multipolar neurons, bipolar neurons, unipolar neurons. Structural classification takes into account the number of extensions arising from the cell body. Functional classification. There are three types of neurons: sensory neurons, motor neurons, interneurons. Functional classification takes into account the type of nerve impulse conduction. Structural classification. One in multipolar neurons, they usually have a number of dendrites and one axon. Most of the neurons in the brain and spinal cord are of this type. Two bipolar neurons, they have a single axon and a single dendrite. They are found in the retina, the inner ear and the olfactory area of the brain. Three unipolar neurons, they have a single extension arising from the cell body. This extension branches into two branches a short distance from the cell body. LA classification fonctionnelle. One is sensory neurons, also known as afferent neurons. They transmit information about the environment to the spinal cord and the brain, dendrites connected to sensory receptors. Two interneurons. They are found in the spinal cord and the brain, interpret sensory information, and trigger an appropriate response. They also transmit nerve impulses from one neuron to another. Three motor neurons, also known as efferent neurons. They transmit impulses or commands to the muscles and glands. Myelinization. Nerve fibers, most commonly axons, and much less frequently dendrites, are sometimes covered by an insulating myelin sheath consisting of a spiral of closely wound layers of plasma membrane that is particularly rich in lipids, cholesterol, phospholipids, glycolipids, which enables them to increase the speed of the electrical signals they carry. We can therefore distinguish between unmyelinated fibers, lacking myelin, with a conduction velocity not exceeding 2.3 meters per second. Myelinated fibers whose conduction velocity, which is proportional to the thickness of the myelin sheath, can reach 120 meters per second. The process of myelination begins during fetal development and continues for several years after birth, which explains why certain functions of the nervous system do not reach maturity until later. Conversely, a process of demyelination may occur during a person's lifetime, leading to multiple sclerosis, MS, a neurodegenerative disease involving glial cells. Myelin production is in fact carried out by oligodendrocytes at the central and by Schwann cells at the peripheral. In the neural axis, CNS, each oligodendrocyte extends several membrane processes in order to myelinate the axons, several dozen, located in its vicinity. As a result, the myelin sheath is regularly interrupted, leaving the axon exposed in short regions known as Ranvier's nodes. In nerves, the process of myelination is slightly different. Each Schwann cell begins by completely enveloping a section of nerve fiber with several turns of its membrane. The myelin sheath is therefore, once again, regularly interrupted by Ranvier's nodes, with the interval between each node corresponding to the area of myelination of a Schwann cell. The synapse and synaptic transmission. For a long time, the very structure of the nervous system was a matter of debate, with two theories pitted against one another. A network-based model, according to which neurons formed a vast network, in which they were interconnected with one another. A neuronist conception, according to which neurons were independent of one another and were connected only by contiguity. The controversy was finally resolved with the advent of the electron microscope in the 1950s, which clearly demonstrated that neurons are independent cells, and that they communicate with one another at specific sites known as synapses. A synapse is a specialized junction through which the nerve impulse nervue is transmissed d'un neuron à l'autre, transmission synaptic, via des messages chimiques, les neurotransmetters. All interneuronal synapses, also known as neuroneuronal, invariably have the same basic structure. A presynaptic element belonging to a first neuron, it is an axonal terminal that forms a small swelling or terminal button. It contains mitochondria and synaptic vesicles containing neurotransmitters. A synaptic cleft which separates the two neurons. This is where neurotransmitters pass from the presynaptic terminal to the postsynaptic terminal, which is on average 20 to 50 nanometers away. A postsynaptic element belonging to a second neuron. It has receptors on its membrane for the neurotransmitters released from the vesicles of the presynaptic terminal. Types of synapses. However, based on morphological and functional criteria, we are led to make several distinctions. From a morphological point of view, we distinguish between axodendritic synapses, between the axon of one neuron and the dendrite of another. Axoaxonic synapses, between two axons. Much less commonly, synapses, somatosomatic, somatodendritic and dendrodendritic. From a physiological point of view, a distinction is made between synapses electrical, chemical synapses chimiques. Synaptic transmission involves the following stages. Synaptic transmission. 1. An action potential reaches the axon terminal. 2. The influx of calcium, Ca+, causes the synaptic vesicles containing the neurotransmitter to fuse with the presynaptic membrane. 3. The neurotransmitter is released into the synaptic cleft via exocytosis. 4. The neurotransmitter diffuses across the synaptic cleft to the postsynaptic membrane and binds to specific receptors located on that membrane. 5. The permeability of the postsynaptic membrane is altered, which generates an action potential in the postsynaptic nerve fiber. 6. The neurotransmitter is cleared from the synapse. The arrival of an action potential at the presynaptic terminal triggers the opening of voltage-gated calcium channels. He then enters the cell. The presence of calcium inside the cell triggers the migration of synaptic vesicles to the membrane. Once they come into contact with the membrane, they release their contents via exocytosis, resulting in the release of the neurotransmitter into the synaptic cleft. As the neurotransmitter has a high affinity for the receptors on the postsynaptic membrane, it binds to them via steric complementarity. Receptors are chemical-ligated channel proteins. In other words, their opening depends on the presence of a chemical substance, in this case a neurotransmitter. Depending on the type of synapse in question, the neurotransmitter-receptor combination results in an excitatory or inhibitory effect on the postsynaptic membrane. Once the action potential has passed into the postsynaptic element, the neurotransmitter is deactivated to prevent the excitation or inhibition it caused by binding to the postsynaptic receptors from continuing. Depending on the type of neurotransmitter, two mechanisms are responsible for this inactivation. Either it is broken down by a specific enzyme in the synaptic cleft. Either it is reuptaken, the most common scenario, by the presynaptic terminal, and can thus be reused.